Hi listeners, welcome back to our theoretical biophysics podcast. Today I have the pleasure to interview Severin Schink, who is in Harvard Medical School currently. And I'm very glad to meet him at the Microbial Physiology conference in Copenhagen we are attending together. So thank you very much, Severin, for taking the time to explain your work. And I'm glad to have this small discussion with you.
Thanks, it's a pleasure to be here.
So what are your interests currently?
I want to find out why bacteria die when they starve. And it's a question that you would think microbiologists have figured out long ago, but it's kind of it's still like one of the big mysteries is we don't really know why bacteria need food to survive, and for some reason some bacteria actually don't even need food to survive, so they can make spores and they can live like literally millions of years. So it's really a mystery to me, and basically I've been trying to figure this out for the last 8 years of my life.
All right, so I see that you've done a terrific work on the death of bacteria, and so can you tell us a bit more about the experiment you've done to investigate this phase of life?
Yeah, so the idea is that okay, so what is actually being alive for a bacterium? So this it's of course a very philosophical question, and I don't want to get too much into it, but essentially, like the definition that we'll work with now is if a bacterium can replicate again, then we call it alive. And so basically, the experiment that we do is we take whatever bacteria culture we have, we dilute it, we put it on an agar plate, and if it is able to grow again, then it makes a little colony, and that colony we can count. And then basically what we do is, for example, we grow our bacteria, then we wash them, we put them into an environment that doesn't have the carbon where they get their energy from, and then over the next like 10 days or something, we measure how many of them can still form a colony.
So you put them in this not so good environment for them, so they are keen to die in this kind of situation, and so what you see is that you have less and less spot on your agar plates with time.
Yeah.
Okay.
So it decreases over time, and now here's the thing that struck me most, and that is when you plot the viability over time on a log axis, it's a perfect line, meaning they die exponentially over time, and this really, the first time I saw this, it really I found it really striking, because when you're growing bacteria, then bacteria grow exponentially. It's like one bacteria making two bacteria, making four, making eight. And it's very intuitive. Like we all know, like this is supposed to give us exponential growth. But when you look at starvation, why are 8 bacteria becoming 4 and then becoming 2 and then becoming 1? Right?
Like weird.
Like it's really weird. Like there should be some it's almost like a physics law, and there should be some like simple explanation behind it. That's what I thought 8 years ago. And so the first thing we thought is that it looks like a Poisson process. So it's like the simplest physics process that you can imagine. It's basically all the bacteria would be independent of each other. They would be all identical, and every one of them dies just because of a random process, and that should give you an exponential decay. Just like, for example, radioactive decay gives you an exponential decay. But it turns out that that's not true. And
what happened?
Things are more complicated. And so we tested, like, there's basically one of the core assumptions of a Poisson process is that the bacteria are independent of each other. They're dying independent of each other. And so imagine you now take a part of your culture and you kill them. You take it, you irradiate it with UV light, or you heat it up, or you grind it into paste, and you put it back. Then you actually see that the ones that are surviving, the ones that you didn't kill, can survive on the ones that you killed.
So cannibalism.
So they're cannibals, right? And that means they're not independent of each other. So it's not a process.
So how can you explain this exponential decay if it's not a process?
So the idea is that basically from that experiment we know that there's always a source of nutrients, right? Every time some bacteria are dying, they're getting supplied with fresh nutrients, and they can do that, use that to do something. And so the idea was, okay, basically from that we learned that they're not inactive, even if they're like, you look under a microscope, they really don't do anything. Like,
they
don't
divide.
wiggle around, they don't divide. But from that we actually know that they must be doing a little bit. So what is that little bit that they're doing, and why is that little bit so important?
So that's super interesting because they are in stationary phase, more or less dying, all of the others, but they are still active in fact, and they have this activity because they've got the food coming from the dead cell around them. Okay, so what do they do in the end with this?
Okay, so one of the things we did to solve that is to look under a microscope. Like, how does a bacterium look like when it's dying? Like, is there something that we can see? And what we found is when you look under a microscope and you look very carefully, that they actually pop. So there's a little bit of a twitch going on in them, like a twitch of death, where they expand a little bit. You can see there's pressure building up in them, and then shortly afterwards you see that they're contracting again. So meaning that the pressure has somehow equilibrated, and so we started working with different dyes that can stain different processes. For example, we use a lot of dye that measures the polarization of the cell, and we can see, okay, right before this twitching, that stain turns on, right? So meaning that, okay, the cells depolarize and then they swell, and then there's another stain that we use, and that goes into the cell when its membrane has a hole.
Okay.
And we saw that, after this expansion that we see, it gets a hole and then kind of like bleeds out a little bit, and then it, then the corpse is just flying around. And
okay, so if I sum up, so first you see the depolarization, so the electric charge in the cell is changed.
Yeah.
And after you see a hole in one of the pore or the membrane, and this leaves some cytoplasm, the possibility to leave the cell, and then this is when they die, actually, right?
So I think this is the point where they die, and like the reason why I think that is that when they, like, somehow death is like this irreversible process. It's like it's the point of no return, right? So if the cell's a little bit injured, it could go back. But if it really loses like a significant part of its like biomass and has a hole in the membrane and just leaks out, then I don't really see how the cell could recover from that. So that's why I think this is actually the death process that makes this this entire that kills the cell. Yeah.
But that still doesn't explain this exponential decay when they feed on each other.
Yeah. So exactly. So we now know they're eating, right? They're constantly eating something, and we know that eventually they'll pop. So then we scratched our heads for a little bit, and then a couple of years later. So, but jokes aside, so the thing is, like, why would a cell pop, right? Yeah. So we're talking about a pressure here, and it's basically like a pressure inside a membrane. It must be like an osmotic pressure. So it's basically that the cell is losing the control of its osmoregulation, and then water comes into the cell, and then pressure builds up, and then it pops. And the reason for that is that every cell, so not just bacteria, but kind of all cells, are packed with biomass, like proteins and RNA, metabolites. There's a lot of counter ions, and all of that stuff is osmotically active. So when the cell kind of pump out ions anymore from the surrounding medium, then they will come in, and then you will have like a net internally more than outside. Basically, if you let the system equilibrate to a Gibbstonian equilibrium, you'll have a positive pressure, and then you get this expansion. And this is, for example, what you see in higher cells in necrosis. So when higher cell dies, it just like slowly expands and like blips off into little pieces. So it's really it's a very general phenomenon that, you know, just cells that don't have any control anymore pop.
So the ion concentration around the cell, in the medium and inside the cell, is very important, and so they've got these active pores which are able to equilibrate them. So how are they involved in the death process in the end?
Yeah, so how the cell can basically prevent itself from popping, right? If like popping is like, you know, you stop doing anything and the cell pops, so then I mean it's kind of like the cell that wants to stay alive and needs to do some pumping of ions. And basically whatever diffuses into the cell needs to get pumped out, right?
That's active.
So that is against the chemical gradient, so it must be active, and it must consume, you know, it must dissipate free energy, and, you know, going back to the beginning, we take away their food, right? So no nutrient, they cannot derive any more energy. You know, they can have some storage, right? Or they can, you know, they can eat up themselves a bit, but, you know, eventually this is going to, any food source is going to run out. You know, they cannot pump anymore, and then you get this expansion. So that's the idea. And then what we thought is, okay, can we formulate that idea mathematically? Can it give us the exponential decay? Can it explain to us, you know, like why does this process look so random? You know, is it random? And yeah, so we built a model, and basically in the model, it's about, like, the one part that is really important is the nutrient recycling, right? So cells die, they spill out their guts, you know, the other bacteria can use it up, and then they use it to pump, and kind of know that, okay, because of the balance of the ions and the homeostasis, basically what they pump must equal what they pump out in order to remain. What they pump out must equal what comes in, right? And that's when you can use, you know, like physics law of diffusion, like Fick's law, and permeabilities and ion gradients, and kind of describe the dynamics of the process.
So what I see here on the paper you are presenting during this podcast is, so there's two sets of equation. One set of describing the nutrients, and another one describing the solutes in the cell. So the solutes are the ions. And so, as you just said, the equation of the nutrients has a source term, which is the release of nutrient by cell lysis, and a consumption term, sink term, which is when cells consume it, so they take it out of the environment. And so there is the second equation on the solutes inside the cell, with diffusing flux from outside to inside of the cell, and active pumping to have this equilibrium and noise. Okay, and so how these actually two equations, which describe nicely and wrap up the physics of the physiology of the cell that happens during their death, the stationary phase, comes to explain actually the exponential decay of the viability of the cells. Can you tell us a bit more what is at play in this process? And for instance, maybe one question is, what is actually setting the exponential decay rate of this?
Yeah. Okay. So one of the observations that really confused us for a while is that when you look at a cell in starvation, it actually seems to be pumping ions really, really well. So the kind of like this is all E. coli. I don't know if we already said that, but it's like E. coli has actually two membranes, and the ion gradient is across the inner membrane. And what you see is that the inner membrane actually contracts, so meaning that they apparently are really good at pumping out ions
Mhm.
and basically increase their internal solute number, right? So that the volume decreases, so that the concentrations, the osmolarity is met. So they're all in this contracted state, which means they pump really, really well. So we somehow needed to build that into the model and kind of understand, okay, why is it that they all seem to be doing so well and then suddenly lyse?
So if I make a stop on this key point, so
Yeah.
the cells are so good at pumping out all the solute and ions they've got inside them that their inner membrane actually shrink a bit compared to the outer membrane. And so they've got this free space in between the inner and out membrane. And this plays a role into the protection against lysis?
Yeah, so if they kind of like this contraction is the opposite of this expansion and lysis.
So they want to avoid dying, they want to avoid expanding.
Exactly.
Okay.
But instead of just, pumping a little bit and, not shrinking, they pump so much that they shrink.
It's super efficient.
So it's like, it's really, really confusing. But then at some point we started realizing that membranes are actually very sensitive to being stretched. Because if you stretch a membrane, like beyond, what material you have, it actually becomes permeable. And if you think, for example, about a boxer on a boxing mat, right? And that boxer is getting like a really hard hit on his head. And basically what's happened is that the brain is getting accelerated and the neurons are getting stretched. And then the membranes become permeable, the ion gradients in the neurons equilibrate, and then the boxer is knocked out. So I think something similar is actually happening here with the bacteria when they want to prevent their membranes from getting stretched, because if they get stretched a little bit, then they become like super leaky. So, and this gives us a positive feedback loop. Basically, if by chance, you stretch your membrane, and more ions come in, then you stretch it even more, and then you have this like runaway process
and
that
this is
goes
catastrophic.
into lysis. And this is like the catastrophe of the cell that it wants to prevent, right? So that's why it pumps as much as possible. And we can put this in the model and basically have this like stretch dependent permeability, and then basically what we can then do with the two equations in the papers. We map it onto like a potential or pseudopotential. So basically we're using a physics trick from stochastic processes. It's called a Paramus escape. So basically, imagine this: you have a stochastic process. You have like a variable and a potential, and the potential has a little well, and your particle is sitting in the well. Then what is the rate at which you hop over the barrier? Right, and we can actually use this, and what we see is that the rate at which they hop over, basically they go from lysis to catastrophic failure and lysis, sorry, from plasmolysis, from the contraction, from the contraction to the catastrophic failure.
Yeah, the lysis.
That rate gives you the height of the barrier, because the barrier is basically like the pumping that protects the cell.
Okay, so because the more cell that lies, the more cell can cannibalize them, and so they are even more efficient to pump out and be protecting themselves against lysing. So it means that if there is a protection barrier for them to get to the lysis state is higher. Okay.
Yeah. So it's like exactly. Yeah. So cannibalizing always gives them like more nutrients, and if a lot of cells die, then the other ones can survive better. It gives you this like this feedback. And what I think is really striking about it is it pushes your entire population of bacteria into like a collective steady state. So basically, and then this steady state is basically the reason why you have the exponential decay. Right? So every day, half of your bacteria die, because they randomly hop over the barrier, and then they supply the rest of the bacteria. And this thing happens on day 1, and on day 10, and on day 5, and so on. So it really it's like this continuous steady state.
Okay. Yeah. Okay. So you've got in this steady state, these two forces, one which is okay, they are starved, so they are less and less efficient to pump out and protect themselves against lysis. But on the other way, there is this lysed cells, this food available, which actually feeds the one which wants to protect themselves. So you got this equilibrium between cells that are going to die because they lack food, but cells that are going to survive because you give them food by cannibalizing the cell around you. Okay. And so you got this equilibrium that stays over a certain amount of time. I mean, it can last days. That's very impressive. Okay.
Yeah, so it goes over like 2-3 orders of magnitude. So from basically the first 99.9% of the bacteria that die, and at some point you have like different effects. Like there's a lot of biomass that they cannot really use, and then you have mutants that can use it, and then you get like overgrowth of mutants. So you get it basically
mutation can happen in this context.
Later, right? So after like 99.9% are dead.
Yeah.
Then you get like mutants popping up, and then actually it's like you can get into a phase of like where they like one after the other like takes over, and like this entire like survival can take them years.
Wow.
Like I don't think anybody ever managed to just let the bacteria sit in a test tube and actually go extinct.
Yeah.
There's like there's like a sequential like overgrowth and mutations. So there's a lot more going on, but we're describing the first 99.9%. So
which is already a lot.
Yeah.
But it's always tempting to go to the extreme case in the asymptotic in the asymptote of the curve and see what happens there.
Yeah.
But definitely the 99% is there. So every day you got half of the cells that dies, and that sets the rate. Actually, is it really half of the cells, or could it be more quantitative?
So that actually depends exactly on what your bacteria are and in what medium they're in. Right? So imagine this: it's actually half for like the experiment that I usually do. That is then because then the experiment is done in a week. But you can actually have them die faster or have them die slower. There's like you can culture them differently, or you can have, for example, more ions in medium, then you need to pump more, then more cells need to die to sustain that pumping, and then gives you a high death rate. So there's all sorts of perturbations that you can do. For example, you can also look at scaling, like how should the death rate then depend on the geometry of a cell, right? So because you have your diffusion into the cell going via the surface, but the nutrients that a dead cell releases is in the volume. So you actually expect the death rate to scale with a surface to volume ratio. You can do that. You can make like tiny little round cells, or like really long rods, or so you can play around with those parameters.
Oh, that's amazing. So okay, the death rate is going to depend on a lot of physical factors. Actually, the cell shape, I didn't thought about it, but this is really striking. Yeah.
It's death as a physics process.
Yeah, in the end, when things stops, yeah.
There's another thing that you can do, and that is, if pumping ions is really so important to the cell, then can you just help it out and just have it not need to pump? And so imagine you take your medium and you remove all the ions that are in there, and you replace it with something that can still do the function of like your regular buffer, like buffer pH, and like be an osmolyte so that it can balance osmolarity. Then you would expect that they don't need to pump anymore, right? So they can retire basically, right? Yeah. It's like no more work needed. And this is actually what we see. We can decrease the death rate and by like 80% or something, and really protect them from dying. And in addition to just removing this cost of pumping, we also see that even if they like eventually they run out of energy to do anything, so they also depolarize, like stuff equilibrates, but they don't last because
they don't have this pressure.
They don't have the pressure problem, right? So they actually just stay there as like an inactive cell. And
you could store them like that.
I think you could store them, yeah. So like my dream would be to go, instead of protecting them 80%, protect them 100%, and then like you can just instead of having to go to like the deep freezer minus 80 degrees, you just keep your bacteria on the bench, like in the old days, but but without like all the mutations accumulating.
Okay, I think time is flying, and it was a really interesting journey in the death of bacterial cells. So thank you very much, Severin, for taking the time to explain and enlighten us with what you found in your quantitative experiment. I invite you to check out the work of Severin. A lot of things on bioRxiv. I really like this system of having the science available for everybody on bioRxiv. And Severin has a super good contribution you can look at. So thanks again, Severin.
Thanks for having me.
And to us listener, I see you next time in another episode.